Let's study general parasitology. So classification of parasites, two major groups, protozoa and helminths. So protozoa can be amoebae, flagellates, sporozoa. So protozoa unicellular, so amoeba, Entamoeba histolytica, Naegleria, Acanthamoeba. Flagellates like Giardia, Trichomonas, vaginalis. Blood parasites like Leishmania, Trypanosoma. Then sporozoa, that is Plasmodium babicea. Other like Toxoplasma, Isospora, Cryptosporidium and Melantidium. Helminths are multicellular. So first is platyhelminths. So they are flat and segmented. Cystodes tape-like. Example, Tinea, Hymenoleupisthana, Diphyllobothrium and Echinococcus. Trematodes leaf-like, unsegmented. So cystosoma. Nemathelminths, nematodes cylindrical and unsegmented. Intestinal nematodes, Ascaris, Ankylostoma, Trichuris, Enterobius, Trichinella, Strongyloids, and tissue nematodes, Wucheria and Dracunculus. Loa loa. Lab diagnosis. So first specimen can be stool or rectal swab, then demonstration of parasitic forms by microscopy, culture, animal inoculation, xenodiagnosis, immunodiagnosis, that is skin test, serological test, molecular diagnosis, DNA, PCR, other laboratory tests to detect cytological or biochemical alterations in blood like CBC, IgE.
Now let's study indications of examination of stool. So first it is required for the detection of parasites, infestation of intestine like hookworm, roundworm, Entamoeba histolytica, which causes amoebic dysentery. Then bacterial infection of intestine studied by cultural examination of stool like bacillary dysentery, which caused by Shigella, enteric fever, cholera. Then chemical examination of stool is done to detect defect in absorption, that is malabsorption syndrome. Urobilinogen estimation to study excessive hemolysis. Occult blood test is done for detection of ulcerative lesions of upper gastrointestinal tract. For lab diagnosis of parasitic infection, the specimen is stool and it is done for detection of ova, cysts, tropozoites, eggs, larvae of the parasites. So collection of sample is done by either direct stool examination or other samples can be NIH swab, that is National Institute of Health, is used to collect samples from the perianal area in the infection of enterobius infection. Then cellophane tape is applied to the perianal area of the patient early in the morning, or string test for duodenal samples for tropozoites of Giardia lamblia. So these are different methods of the sample collection of the stool. Then after that, stool preservation can be done in polyvinyl alcohol, merthiolate iodine, Shodin fixative or formalin solution. So this is a stool container. In that plastic container, sterile container with the spoon attached to the lid of the container. Now when to examine the stool sample? So if consistency is liquid stool, then you have to examine it within 30 minutes, because in that we are examining tropozoites, that is motility of the parasites. So tropozoites like amoeba or giardia. Then semi-solid stool we can examine within 1 hour, and solid stools of formed stools up to 24 hours after collection, because in that we see mainly cyst forms. In semi-solid we can see both tropozoite and cyst form. The first step is macroscopic examination of stool. So consistency of stool, like it is liquid, semi-solid or solid. Then blood or mucus present or absent. Then few parts of the intestinal parasites like adult segments like Ascaris, Enterobius vermicularis, tapeworm. So we can see that parts of the parasites. In microscopic examination of stool, first step is the routine examination, that is by saline wet mount and iodine wet mount. So in saline mount, procedure is take 1 drop of normal saline on slide, mix stool sample, put cover slip and see under microscope. The advantages of saline mount: useful in detection of tropozoite cyst of protozoa, eggs and larvae of helminths. Motility of tropozoites and larvae can be appreciated. Bile staining property, that is they appear golden brown, and non-bile stain eggs appear colorless. So non-bile stain eggs examples are hookworm, enterobius and H. nana, HEN. Then iodine mount, the procedure, take 1 drop of Lugon's iodine on slide, mix stool sample, put cover slip and see under microscope. Advantages: the nuclear details of the protozoan cyst, helminth, eggs and larvae are better visualized. Disadvantages: iodine immobilizes and kills the parasites, so motility of the tropozoites and helminth larvae are not appreciated, and bile staining property is not appreciated. Second method is the permanent stain smears. So it is done for detection of protozoan cyst and tropozoites by staining their internal structures. The common methods are: first is the iron hematoxylin stain or trichrome stain and modified acid fast stain. It is useful for the coccidian parasites like Cryptosporidium and Isospora. So in that 0.5% acid, that is H2SO4, is used, and that is why it is called modified acid fast stain. And these parasites are mostly opportunistic infections in the HIV patient. When eggs or larvae or parasites are very few in number, then concentration techniques are used. So first is sedimentation method and second is flotation method. So in sedimentation method, the parasites are recovered in the sediment using either gravity or centrifugation. So the techniques used are formalin ether or formalin acetone. So stool sample and these reagents are taken in centrifuge tube and then it is centrifuged and then deposit is taken. In that we can find the morphology of the parasites. The advantages of sedimentation method is easy and minimal technical error is there. Morphology of eggs and cysts are preserved and it is more sensitive. The flotation techniques, stool is mixed with solution having high specific gravity, and the techniques used are saturated salt solution or zinc sulfate flotation method or sugar flotation method or saturated magnesium sulfate solution techniques. The disadvantages: some helminth eggs like trematodes, tinea, unfertilized eggs of ascaris do not float in the salt solution, and then distortion of morphology of the eggs and cyst is also there. So in this stool microscopic examination, we can see different morphological forms of the parasites. This is stool microscopic examination. So in that we can see different morphological forms. So first is cyst of Entaminma histolytica. In that we can see 4 nucleus. Then cyst of Giardia lamblia. In that we can see 4 nucleus, but the cyst is usually oval. Then tropozoite of the lamblia. So in that we can see flagellas. Then egg of H. nana. In that we can see the circular egg and it with the flagella. Then in tinea we can see it is circular usually, and we can see 3 pairs of hooklets and the transverse striations. Then egg of hookworm, in that we can see 4 blastomers. Then fertilized egg of Ascaris lumbricoides. So here you can see space between the covering and then the center is the embryo. Unfertilized egg of Ascaris lumbricoides. So here you can see this no space and it is full of the dots and covering. Then egg of vermicularis, Enterobius vermicularis, it is mostly by one side convex and one side flat, and then here you can see in the center embryo. The egg of Trichuris trichuria, so it is usually embryo inside and then covering of double layer and 2 mucus plugs at the both ends.
Hello students, let's study protozoa. So in that amoeba, Entaminma histolytica, flagellate Giardia trichomonas, blood parasites Leishmania trypanosoma, sporozoa, plasmodium. First, Entaminma histolytica, host man, infective form, mature quadrinucleated cyst, mode of transmission fecal-oral, clinical manifestations, amoebic dysentery, passage of blood and mucus in stool, amoebic liver abscess is the complication. Lab diagnosis, stool microscopy, tropozoites and quadrinucleated cyst. Giardia lamblia, host man, infective form cyst, mode of transmission fecal-oral, clinical manifestation diarrhea, pain in abdomen, fever, malabsorption of fat, steatorrhea, passage of fat in stool, chronic enteritis, enterocolitis. Lab diagnosis, stool microscopy, tropozoites and cyst. So tropozoite you can see motility in saline preparation and cyst you can see in iodine preparation. Third is the Leishmania dononi, infective form promastigotes, Vector sandfly, transmission bite of the sandfly, clinical manifestation fever and hyperpigmentation, splenomegaly, lymphadenopathy, pedal and ascites, hematological abnormalities, pancytopenia, anemia, leukopenia, thrombocytopenia. Lab diagnosis, specimen blood, pancytopenia, and PBS LD bodies. That is a mastigote form of the Leishmania dononi. Plasmodium species Plasmodium vivax, Falciparum ovale, malaria, knowlesi. Life cycle, mode of transmission, bite of mosquito, female Anopheles mosquito. Infecting agent, sporozoite. Human pre-erythrocytic schizogony, merozoite zygont. Erythrocytic schizogony, merozoite zygont. Gametogony, male female gametocytes. Mosquito sporogony, microgamete plus macrogamete, form zygote, then oocyst, then kinate, and then the sporozoite. Pathogenicity, malaria. Febrile stage, cold stage, hot stage, sweating stage. Anemia, microcytic hypochromic because of the breaking down of RBCs during segmentation of parasites. Third, splenomegaly, enlargement of spleen. Complications of P. falciparum, black water fever, pernicious malaria, cerebral malaria, algae malaria, septicemic malaria, hypoglycemia, bleeding, renal failure, pulmonary edema, jaundice. In Vivax, it is relapse. Lab diagnosis, specimen blood, PVS, thick smear, screening, thin smear, species identification. So morphological forms, ring form, tropozoite, zygont, gametocytes, four forms seen in P. vivax and ring form and gametocyte form seen in P. falciparum. Quantitative Buffy coat examination, rapid malaria antigen test.
So, this malaria, I am Dr. Rajeshri Mutha, Department of Microbiology, SMPT, IMS and RC. So the name malaria is mal means bad and area means air. So this is spread by air pollution through stagnant water and marshy lands. So the Alphonse Lavrin discovered causative agent of Plasmodium RBC of a patient in Algeria in 1888, and Sir Ronald Ross in 1897 discovered sexual cycle of parasite in mosquito in Secunderabad in India, and both got Nobel Prize for the contribution to malaria. The causative agents of malaria, previously they were four, now it is five. So first is P. vivax, it causes benign tertian malaria. So benign tertian means it is benign, so not very complicated, and tertian means the periodicity of fever is every 48 hours, that is every third day. P. falciparum, malignant tertian, so periodicity of the fever is every third day. Then, but it is with the complications. Then P. malariae, so benign cotton malaria, so fever spike is every fourth day, that is 72 hours. P. oval, it is similar to vivax, that is benign tertian malaria, and the fifth species new one is added, that is P. knowlesi, so it is quotidian or simian malaria, so every alternate day fever, that is once in a 24 hours. So it is parasite of monkey but can infect humans also. So it is considered as a fifth agent. Now, transmission and life cycle of malaria. So definitely host is female Anopheles mosquito because sexual cycle, that is sporogony, takes place in the mosquito. And intermediate host is man because asexual cycle, that is schizogony, takes place in the man. Infective form, the sporozoites. Those sporozoites enter man after the bite of mosquito and the life cycle continues. Portal of entry is bite through the skin. Mode of transmission, bite of the infected mosquito, and site is first in the liver cells and then in the RBCs. The life cycle, first mode of transmission is bite of the female Anopheles mosquito, you already saw, and rarely it can be through the blood in blood transfusion or transplacental forms. In that is infective form is tropozoites or merozoites. So this picture is showing you life cycle of malaria. So here two phases are there, one asexual phase in man and then sexual phase in mosquito. So in man there are mainly two stages, that is pre-erythrocytic schizogony and erythrocytic schizogony, and in vivax and oval you can see exoerythrocytic schizogony, and in mosquito after that it is gametogony, and then in mosquito it is sporogony. So we will see it in detail. So in this picture, we will see the two cycles in man and mosquito in detail. So first, after the bite, sporozoite enter the
skin, and then through the blood circulation they go to liver cells. There they develop into the zygont, that is merozoites, and then merozoites develop into the tropozoite and zygont. So this is called pre-erythrocytic zygogony or pre-erythrocytic zygont formation. Then some merozoites in vivax and oval, they go in dormant stage. They are called as hypnozoites, and after some days or months or years, again they may cause infection. That is called as relapse. Then the merozoites, after zygont ruptures, the merozoites are released and then they infect RBCs. Again, merozoites will develop into tropozoite zygont, and it is called erythrocytic zygont containing merozoites. So again, merozoite will rupture and then the zygont will rupture and merozoites are released. So these merozoites will again reinfect RBCs, or some of them may convert to the sexual phase, that is gametocytes. So male gametocytes and female gametocytes. So male gametocyte is called as microgametocyte and female is called as macrogametocyte. So at this stage again the mosquito will bite, and these gametocytes, male and female, they will enter into the mosquito. Now cycle in mosquito, it is called as sporogony. So these the micro and macro gametocytes, they will develop into macrogamete and microgamete. So microgamete is having around 8 flagella. So it undergoes X flagellation and each one forms the microgamete, and then it will fertilize with macrogamete, and then it will form zygote. Then from zygote it will form ookinete. Then ookinete, it will develop into oocyst in the midgut of the mosquito, and then oocyst containing sporozoites will reach to salivary glands. Then oocyst will rupture and then all the sporozoites are liberated in the salivary gland, and with the bite to the humans, this sporozoid form, that is infective form, is entering the human skin. So this is all about cycle of plasmodium in human, that is zygogony, gametogony, and in mosquito it is sporogony. So few dormant forms will that is hypnozoites, again they cause relapse, and that is called as exoerythrocytic zygogony. So already saw in human cycle there are mainly two: pre-erythrocytic zygogony, erythrocytic zygogony, then gametogony, and in vivax and oval you may see exoerythrocytic zygogony, that is hypnozoites will again mature into the zygont form with merozoites. In mosquito it is gametogony. Sorry, in mosquito it is sporogony and the development of the gametocytes into gamete and fertilization, and then again the infective form sporozoite is formed. Now, what are the morphological forms seen in humans? So, in P. vivax, it is mainly ring form, it is thick and single. Then, tropozoid form, it is large, irregular, amoeboid. Then, third, zygont form, it is large, filling completely inside the RBCs and containing merozoites. Then, gametocyte, it is spherical, light blue cytoplasm. And then, RBCs are enlarged with the Schaffner's dots present in the RBC in P. vivax species. In P. falciparum, mainly it is ring form, which is thin or delicate, and many sometimes multiple rings can be seen inside the RBCs, and sometimes you can see headphone appearance or cold forms. Then tropozoite is not seen, zygont is not seen in P. falciparum. Then gametocyte, they are banana shaped or crescent shaped, and they are having pink cytoplasm. RBCs are usually normal and they contain Morris dots. So these are the pictures of P. vivax, ring form, tropozoite, zygont and gametocytes you can see in the PBS. Then these are the Plasmodium falciparum ring form and gametocyte form you can see inside the RBCs. And then in P. malari, especially it is late tropozoite showing band form. Then P. oval, it is RBCs are oval, and sometimes they are crenated. So pathogenesis. So there is mainly triad of fever, anemia and splenomegaly. So fever is because of the liberation of merozoites in the RBCs. Then anemia because of the destruction of erythrocytes, and splenomegaly because of the hemolysis, rapid hemolysis. The RBCs go to spleen and then spleen enlarge in size.
So mainly triad of these three. Then fever mainly consists of first cold stage. Patient will feel shivering and rigors. Then hot stage, so that is increased temperature, and then sweating stage. Complications of malaria is seen mainly in P. falciparum. They are also called as cerebral malaria or black water fever. In Vivax, it is sometimes Vivax and oval showing relapse because of the exoerythrocytic schizogony. Then P. malaria and knowlesi, they are mainly causing nephrotic syndrome, and chronic complications of malaria, it is tropical splenomegaly syndrome. So malignant malaria, what are the complications? So these are cerebral malaria. This we will see in detail. Then pernicious malaria, that is, it consists of black water fever, algid malaria, septicemic malaria, and the other systems are also involved. So RS, it's showing pulmonary edema or adult respiratory distress syndrome. Then sugar is low, so hypoglycemia. Then kidney is involved, that is renal failure, bleeding, diathesis. Then severe jaundice, anemia, and acidosis. So we'll see cerebral malaria. So it is due to plugging of brain capillaries by sequestrated parasitized RBCs leading to vascular occlusion and cerebral anosmia. So here in this picture you can see these RBCs are getting attached to the endothelium of the capillaries, and then because of that sequestration there is clot formation, and because of that there is hypoxia, decreased oxygen supply to the brain, and then it causes cerebral malaria. So patient will have symptoms like convulsion, and patient may go in coma, or in cerebral malaria there may be death of the patient. So cerebral malaria causes anemia, thrombocytopenia, then encephalopathy, retinal hemorrhages, hepatosplenomegaly, hypoglycemia, then metabolic acidosis, renal dysfunction, and sometimes 15 to 20% death. Then second, pernicious malaria. It is characterized by black water fever, algid malaria, and septicemic malaria. So black water fever means because of the sudden intravascular hemolysis after the fever, there is hemoglobinuria, and the urine is dark in color. That is why it is called as black water fever. And it is mainly seen in P. falciparum. Algid malaria, there is cold clammy skin, then hypotension, peripheral circulatory failure, and shock symptoms. Septicemic malaria, high degree of prostration, high grade fever with dissemination of the parasite to various organs, causing multi-organ failure. So here you can see the urine color is dark, that is seen in hemoglobinuria. It is almost dark brown or black in color. Then patient is having hepatosplenomegaly, so liver and spleen are enlarged. Patient is having jaundice, it is mainly type of hemolytic jaundice, and then patient goes in renal failure. Now we will see laboratory diagnosis of the malaria. So these are divided into two, that is microscopic test and non-microscopic test. So in microscopic test, they are mainly peripheral blood smear, that is gold standard for the diagnosis of the malaria, that is thick smear and thin smear. Then fluorescent microscopy, quantitative Buffy coat examination. And in non-microscopic test, it is antigen detection, and sometimes culture and molecular can be done. Peripheral blood smear. So it is simple and gold standard confirmatory test for the malaria. So specimen is mainly either venous or capillary from the earlobe or from the finger, or in children or infants it is grade 2 or hill. Then time usually after the height, after few hours of the height of the fever, and before taking anti-malarial drugs. And it should be at least twice till you don't see the parasites. So now types of peripheral blood smear, they are thin and thick smears. So in this picture you can see thick and thin smear. So first thin smear should be screened, and if you don't see parasites then you can go for thick film, or in some PSCs or some areas directly they go for the thick film because it is mainly sensitive. So for thick film, it is one large drop you have to take 2 to 3 drops in the center of the slide, and then you have to just make a circle with help of coin or with help of another slide, dry it and then stain it. Thin smear at the edge of the slide, you take 1 to 2 drops of blood, and with help of another spreader slide, you have to spread that drop forward so that you will get tongue shaped smear. So now staining, there are two methods, field stain and Leishman stain. So in field stain, you have to fix the smear after drying. So fix with methanol or with spray, then dry, then put that slide in a few dips into the field B, wash with water, then field A, few dips and then wash with water, dry in air and put oil and see under oil immersion lens. In Leishman stain, you have to flood the smear with Leishman stain. You don't have to fix the smear because the methanol is already present in Leishman. Then buffer added and then wait for 5 minutes. Then wash with water and then dry and see under oil immersion lens after putting oil. So first thick smear. So advantages, it is helpful when the parasite count is very low, 5 to 10 parasites are there, still you can see thick smear. So in thick smear you just see the parasite forms. The RBCs are destructed, so you don't have to see, you don't see RBCs. In thick smear, you can do quantification of parasites and demonstrate malarial pigments like Schaffner's dots or Morer's dots. The disadvantage here, species identification or morphological forms identification is not possible. In thin smear, advantages, it is useful in species identification. All the morphological forms you can demonstrate or the RBC changes you can demonstrate in thin smear. But disadvantage, it is having low sensitivity, that is you need more than 200 parasites per microliter of the blood. So when the parasite count is very low, you cannot, mostly you don't see parasites in the thin smear. So this is picture showing thick film. So here you can see very few RBCs but mainly parasites. You can see dots and nucleus. And the another picture, thin blood smear, you can see RBCs, then nucleus and ring, proper ring form. Whether it is thick, thin, you can see, or other morphological forms you can study in thin smear. So these are all Plasmodium vivax forms, ring form, tropozoid, schizont and gametocytes. Then these are all morphological forms of P. falciparum. Mainly they are ring form and gametocyte, banana shape. Then in oval you see oval RBCs and in P. malari you can see band form of the tropozoid. These are all morphological forms of all the four species. Now, you can go for fluorescent microscopy. So in fluorescent microscopy, you have to just stain the smear with fluorescent dyes, that is acridine orange, and then you can see under fluorescent microscope. So there you can see greenish yellow fluorescence of the parasites. In quantitative Buffy coat, you have to take blood in capillary tube that is coated with the acridine orange fluorescent dye. Then centrifuge that capillary tube, so it will have separation of Buffy coat. So Buffy coat is between junction between the cells, that is RBCs and plasma. So it contains mainly WBCs and lymphocytes. So, and then that you can see under UV light or under the oil immersion lens. So here you can see greenish yellow fluorescence of the parasitic forms. Now second test, nowadays it is performed many times. It is rapid antigen detection test. So here two antigens are used. So for all species, that is pan species, that is it is PLDH, then that is lactose dehydrogenase, and for Plasmodium falciparum it is mainly histidine rich protein 2, that is PFHRP2. So advantages, it is simple, but disadvantages you cannot differentiate other species in pan, and gametocyte stage or other stages are not developed, and it is not developed for the P non-AC. So vector control, for anti-adult you have to use insecticides or you can use bed nets. For anti-larval, larvicide, use of mineral oil or Paris green to kill mosquito in the stagnant water, or biological fish, that is Gambusia fish, can be used to kill the mosquito larva in a stagnant water. You can use that.
Hello students, let's study helminths. Cystode, Tinea hymenopis nana, Diphyllobothrium echinococcus. Trematodes, Cystosoma. Nematodes, intestinal, Ascaris, Ankylostoma, Trichuris, Enterobius, Trichinella, Strongyloids. Tissue nematodes, Wucheria draconculus. Tapeworm, the types Tinea saginata, beef tapeworm. Tinea solium, pork tapeworm. Egg spherical brown in color, and then two linings are there. Thick walled, radially striated with three pairs of hooklets inside. Pathogenicity tinniasis. Increased appetite, loss of weight, abdominal pain, weakness, headache, vomiting, nausea. And second is cysticercosis. Clinical feature depend on cyst location. Subcutaneous cysticercosis, palpable nodules. Muscular cysticercosis, muscle pain, weakness. Ocular cysticercosis, proptosis, diplopia, loss of vision. Neurocysticercosis, brain involvement. Laboratory diagnosis, stool microscopy, gross examination, adult tapeworm and microscopic examination, eggs. Hymenolopis nana, egg spherical oval in shape, two membranes, outer membrane, inner membrane, and three pairs of hooklets and polar filament is there. Pathogenicity, GI symptoms, nausea, vomiting, diarrhea, abdominal pain. Lab diagnosis, stool microscopic examination, H. nana, non-bile stain, egg. Echinococcus granulosus, dog tapeworm, hydatid cyst. Hydatid cyst disease, echinococcus asymptomatic, symptomatic cyst rupture complications, immediate pyrexia, urticaria, itching, skin rash, anaphylactic shock. Lab diagnosis, X-ray, CT scan, MRI to detect hydatid cyst in liver, lung, kidney, brain. Cassonese test, injection of 0.2 cc fluid from hydatid cyst intradermal to patient, immediate Wilhelm flare reaction, positive test. Nematodes, Ascaris lumbricoides, roundworm. Symptoms produced by adult worm, abdominal pain, malnutrition, intestinal obstruction, skin rash, conjunctivitis. Symptoms due to larva, Ascaris pneumonia, symptoms of pneumonia, fever, cough, breathlessness with sputum production, blood tinge, contains Ascaris larva and eosinophilia. Lab diagnosis, stool microscopy, examination, eggs, fertilized, unfertilized eggs. Ankylostomia duodenal hookworm. Worm causes hookworm disease, ankylostomiasis, characterized by anemia, therefore called hookworm anemia. It is microcytic hypochromic anemia. Symptoms abdominal pain, loss of appetite, iron deficiency anemia, ankylostoma dermatitis because of entry of worm through skin when man walks barefoot on fecally contaminated soil. Lab diagnosis, stool microscopy, examination, eggs. Ucheria bancrofti, Ucheriasis, filariasis, chills at midnight, fever, tendering venous lymph nodes, orchitis, epididymitis, complications, obstruction of lymphatics, chyluria, hydrocele, elephantiasis of scrotum, lower limbs, blood, PBS, microfilaria.
Hope you find this session useful for your understanding and for your exams. Thank you.